So this is our first video lecture. This is section 4.1 called Species, Communities, and Ecosystems. So we'll start by reviewing the definition of a species. Here we go. You should know by now that a species is a group of organisms that can potentially interbreed and, importantly, produce fertile offspring. So this horse is an example of a species. Donkeys are another species, because a donkey can reproduce with other donkeys and produce fertile donkey babies. Now, if we take a horse and a donkey and we breed them together, we get what's called a hybrid. Now, a hybrid is not a fertile offspring. Instead, it's one generation which cannot reproduce to make more fertile offspring. So in this case, we have a mule. Now, you can take a horse and a donkey, breed them together, and make a mule. But a mule cannot breed with more mules to produce more offspring. The mule is sterile. It cannot produce offspring of its own. Therefore, the mule is not a species, rather it is a hybrid. Now, most of the time, we do not get new species by hybridization. Rather, new species will usually form by a process called reproductive isolation, which is pretty simple. The idea is that if a population splits off from itself, they will no longer be breeding with each other. In this example here, we have two groups of birds. These are two populations of birds which have split apart from each other across this waterway, and now they are no longer going to reproduce with each other. They are now reproductively isolated from each other. Now, this can eventually, given enough time, lead to two different species of birds. It's important to remember or realize that reproductive isolation is not always due to a physical separation. Think of the lizards in our backyard. These are called anoles. Now, these anoles have this flap. Right here, you may be seeing these. It's called a dewlap. Now, when these lizards mate, they will select a mate based on, the quality, if you will, of the dewlap. And so some lizards will prefer a greenish dewlap. Other anoles will prefer a reddish dewlap. And so this leads to reproductive isolation because the green dewlaps will not breed with the red dewlaps and vice versa. So now we're looking at a separation here, but it's not a physical separation, it's a behavioral separation. This can still lead to reproductive isolation, which could potentially give us new species. Now, when one species breaks off into different groups, we can call these populations. And we'll get back to that in a few slides. So, different species are defined by their behaviors. And so to get started, relating to ecology, these species will have different feeding methods. This is our third understanding. So we can designate a species as either being autotrophic, where they make their own food by photosynthesis or some other chemical reactions, or heterotrophic, where they feed on other organisms for their nutrition. And then sometimes we have strange examples like this Venus flytrap, which can do both. So as far as autotrophic organisms go, we have two broad categories. The first is called a photoautotroph, which basically uses light, hence photo, to make its own organic nutrition. So these are typical plants and algae. Any photosynthesizing organism is a photoautotroph because they're using light energy to drive the production of organic nutrition. A chemoautotroph is an example of an organism which uses oxidation of inorganic compounds to produce food for itself. One example, it would be bacteria living in deep sea thermal vents like this. This is a volcanic vent at the bottom of the ocean. There's lots of heat, lots of inorganic nutrition, and there are little bacteria which can do oxidative reactions to turn that inorganic material into food for itself. Then other slightly larger organisms can go in and eat those bacteria, and we can then get a whole ecosystem of strange creatures living at the bottom of the ocean where no light is present. This is not photosynthesis, it's chemosynthesis. And these bacteria that do this process that starts the whole ecosystem. These are called our chemoautotrophs. Another example of a chemoautotroph would be this bacterial species right here. It's called Rhizobium. And what Rhizobium do is they move into the roots of plants and they form these nodules. And what they do is they help to fix inorganic nitrogen from the air into an organic source of nitrogen that they can use for food. There's also a bonus for the plant, which is that the plant now gets some added nitrogen, which it could not take out of the atmosphere on its own. So there's a nice symbiotic relationship here between the Rhizobium and the plant species. Again, these bacteria that move into the roots and build these nodules, those are chemoautotrophs. They're taking inorganic nitrogen from the atmosphere and making it into organic nitrogen, which they can use and also the plant can use. So they help to support this ecosystem. Now, either way, if we're talking about photoautotrophs or chemoautotrophs, they're going to form the foundation of an ecosystem. So in this waterway, we're going to have some algae, which will then be, fed on by small species, which then start food chains and food webs that support this whole ecosystem that you see here. Same is true for plant species around the waterways here. So autotrophs form the foundations of ecosystems. And then, if we move away from autotrophs, we can get what are called heterotrophs, or in very strange examples, you could have both mixotrophic species, or what these are called. They utilize more than one method of organic nutrition, meaning they can be both autotrophic and heterotrophic at the same time. This Venus flytrap can conduct photosynthesis, so it is a photoautotroph. But, as you can see here, this poor little cricket can be digested by the plant as well, and so it is heterotrophic and autotrophic at the same time. We call this a mixotrophic species. Another example of a mixotrophic species is this dodder, D-O-D-D-E-R, not daughter, but the dodder is a parasitic vine which can conduct photosynthesis, but it also stabs plants with these little barbs as it wraps around it, and it will extract the sap from this plant and eat it. So it's parasitic, heterotrophic, and photosynthetic, autotrophic. So it's another mixotrophic species. IB wants you guys to recognize Euglena as an example of a mixotrophic species. Note the chlorophyll present in green. That's for photosynthesis. There are also little food vacuoles which can then gobble up small bits of things floating around in the water that it lives in. So it's both autotrophic and heterotrophic, another mixotrophic species. Now, if we do have an organism that is strictly eating other materials, not making food for itself, this is what we call a consumer. Consumers must ingest other living things. They're not making any food for themselves. They're strictly heterotrophic. We can break up consumers into some subcategories. Here's an example of a detritivore. Now, basically, detritivores will eat detritus. This is dead or decomposing material and also feces, and they do this through internal digestion, meaning that they must swallow their food and then digest it. Here's your pillbug or roly-poly. You'll find these living under leaves where there's lots of decomposing material in the soil. They'll eat that decomposing material and some feces, and then their excrement becomes part of the nutrition for the overall ecosystem. These are detritivores. Now, they're not decomposers. They're not breaking down materials. The decomposers are what we call in this class a saprotroph. Saprotroph. And they decompose materials using a process of what's called external digestion. Now, if you think about external digestion, this would be like if you held an apple in your hand, and then your hand was capable of digesting that apple. Then you could just absorb the nutrition from the apple. This is what fungi do. These are decomposers. Now, if you look at a mushroom, most of the actual body of the mushroom is hidden underground in a network called its mycelium. The mycelium consists of many root-like fibers, which we call hyphae, and the hyphae will release digestive enzymes outside of the mushroom onto the surface of the wood or whatever it is that they're eating. Those enzymes will digest all the materials out here. Then the mushroom or the fungi rather, can absorb all of that pre-digested material. So this is an external digestive process. Again, these external digesting organisms are called saprotrophs, also referred to as decomposers. Now, when the decomposers interact with our consumers and our producers or autotrophs, we can get a sustainable ecosystem. Which brings us to our next topic. When we're studying ecology, we have to think of different realms or layers of interactions between living things. We've discussed species. If we move up from the species layer, we get what's called a population. Now, population would be members of the same species living in a given area at a given time. So we have a population of flamingos in this image here. Now, if we step up and we add different types of species interacting with each other, we get what's called a community. So this is a community of waterfowl or birds living in the water. We have an egret, we've got an ibis, and a roseate spoonbill. Different species interacting with each other at the same time. This is a community. If we look at this community and the way that it interacts with the non-living or abiotic world, like the water and the air around them, now we're looking at an ecosystem. This includes the biotic communities and the abiotic environment in which they live. And now we're talking about serious ecological study. In ecology, we're looking mostly at ecosystems. So moving on, for an ecosystem to be successful, it needs to cycle all the nutrients present. And the way that works is that autotrophs obtain nutrients from the abiotic environment. Heterotrophs eat those autotrophic organisms, and all of that nutrition can get recycled. All those dead heterotrophs and autotrophs get recycled by saprotrophic decomposers. And so when all this comes together, an ecosystem can be sustainable for long periods of time because producers feed the consumers, which feed more consumers, which can then be broken down as they die by our decomposers. That returns to the soil, and the producers can absorb that nutrition, and everything can recycle over and over and over. And as long as we have a constant supply of energy for our producers, for our autotrophs, then this ecosystem can just keep on going because these nutrients should not ever go away. They should just get turned into different forms, and they can be recycled. And this brings us to our mesocosms. So skill 2 from this section was the mesocosm that you built or designed last week. This is just an example of a sealed, sustainable model of an ecosystem. And so in here, in this bubble with this adorable old man, we have a sealed ecosystem. So all the nutrients stay put, and as long as enough light can get through the glass to feed all these producers in here, then we should continue to have a productive ecosystem because the nutrients continue to get recycled. And this is basically the end of 4.1. I'm going to leave you here. I highly recommend that you read the section. And then your classwork this week, which is posted to Teams, will be to read the last skills about using chi-squared analysis for studying ecosystems and how they interact with each other, or how communities can interact within an ecosystem. So we have DBQ on this process called chi-squared. It's a statistical test to see how strong a relationship is between species. I'd like you to read that section, try the DBQ, and submit your answers on Teams when you finish. That's it. Have a good week.